navigate. Oh, that was loud. Let's try that again. Diagnosed in 2018 with Parkinson's, and when looking for resources, I had been in the nonprofit realm and an advocate for the cancer community. When looking for resources in the Parkinson's community in my area and had trouble, and now being someone that had been in the healthcare system for about 12 years prior, and knowing what systems out there had trouble navigating, I did with the help of from family and friends, decided to start the Great Strong Foundations to be a resource, a local resources for people with Parkinson's. And today I have the honor of presenting Dr. Lee Mosley from the University of Nebraska. And he's going to talk to us about his research with the immune system and T cells in treating Parkinson. Give it over to Dr. Mosley.
Hello. So I'm Lee Mosley. Right now I'm a professor of pharmacology and experimental neuroscience at the University of Nebraska Medical Center in Omaha, Nebraska. I received my bachelor's and master's degree from the University of Oklahoma in microbiology. I then went to the University of Tulsa, where I received my Ph.D. I worked in mucosal immunology. After that, I went to the University of Michigan in the Department of Pathology, and it was a joint postdoctoral fellowship between them and Park Davis. I worked on models of inflammatory bowel disease. Let's see, and after that, I stayed in Michigan for about 6 years as an assistant research professor working in the immunology of aging. And after that, I got an assistant professorship here at the university, and it was in, I was doing aging research and transplantation immunology. And during that time, this program started up with Dr. Gendelman and a graduate student, and they were looking at immune ways to look at Parkinson's disease, look at neurodegeneration. And that's when I decided to come along. And so that's kind of been a it's probably been about a 20-year journey, I believe, or an 18-year journey. I think I came over in 2003. So should I go ahead and start?
Yes.
Okay. Share my screen.
Thank you, Tiffany. I was on mute there.
That's okay. I gotcha.
Can everybody see that?
Yes, sir.
Yes.
Okay. So I guess all of us pretty well know about Parkinson's disease, so I'll kind of cut to the chase. And we're mostly interested in the inflammatory response for the most part, particularly in our models. I'm not going to talk a lot about animal models that we use. I think on the flyer it said that I'd be talking about glatiramer acetate, and that was back 18 years ago when we started that. So I probably won't talk, I'll mention it just briefly, I won't talk about that. But really, not many people realize that inflammation is a major part of the Parkinson's disease. And here you can find in a Parkinson's patient, all of this purple over in here and here are the inflammatory response. These are called microglia. And they're responsible for generating the inflammatory response and kind of cleaning up the system. Well, what happens in Parkinson's is we have this cycle of misfolding and folding, inflammation, and neuron degeneration. So what happens is the neuron starts to degenerate, and alpha-synuclein, which is a protein that's in the neuron, it begins to misfold and accumulate. And pretty soon you get what's known as Lewy bodies. And when the cell dies, it releases all of these alpha-synuclein molecules, and they attach to these resting microglia, which activates them. And once these become activated, then they release neurotoxins and pro-inflammatory cytokines, apoptotic proteins, reactive oxygen species mediators, and all of that spreads then to surrounding neurons. And they become degenerating. Their alpha-synuclein becomes nitrated, it becomes folded. And so it's an non-ending cycle, a non-ending cascade. So what we found is that in our models of Parkinson's disease, if we can attenuate the inflammation, we can spare up to about 90% of the neurons. And this has been done with pharmaceutical reagents. And if we can actually knock out some of these inflammatory mediators, we can actually spare like 90% of the dopaminergic neurons that are destined to die. This is really important, particularly because by the time you're diagnosed with Parkinson's, about 50 to 70% of your neurons are already gone. Well, it also becomes really important from the study that had a very large healthcare cohort, which took NSAIDs for at least 2 times a week, and they found that their incidence of Parkinson's disease was much lower. That actually translated to those that were taking ibuprofen. So our overarching hypothesis here is that if we can attenuate the neuroinflammation, we can protect the dopaminergic neurons from neurodegeneration. And so our strategy is to develop these immune strategies in order to attenuate neuroinflammation and protect neurons. Hopefully, it'll be received a lot better than the Anti-Vaccine Society received Jenner's first vaccine for the cowpox. You can see all these cow parts that are coming out of these people that have been vaccinated. So I guess it's also note that even back in Jenner's time, they even had the anti-vacciners around. Mm. Well, our strategy is looking at regulatory T cell. We're trying to figure out how best to upregulate and boost the regulatory T cell population. And so you may ask, what is the regulatory T cell? Well, these are T cells that actually are responsible for immunological tolerance. These are the cells that tell you not to see you as foreign. If you're seeing yourself, if you're seeing T cells are seeing you as foreign, it's reacting against you. So a lot of these autoimmune inflammatory diseases then are going to be associated with the lack of these regulatory T cells. If you don't have these T cells, you actually will develop very early in life this syndrome called immunodysregulation polyendocrinopathy enteropathy X-linked syndrome, or IPEX syndrome. I guess you see why they call it IPEX. I couldn't say that all the time. But we also know that they're important in experimental models of neurodegeneration and nerve trauma. That's been shown. How do they work? Well, they secrete suppressive or anti-inflammatory cytokines like IL-10, TGF-beta, and IL-23. They inhibit antigen presentation such that the antigen can't be presented to the T cell to actually get it going. And there's a lot of reasons why. There's metabolic disruption by which they can take the CD39 and CD73 molecules. These are exonucleases, and they can convert ATP to a molecule called adenosine. This adenosine is then a ligand for the A2A receptor, and that sends a negative message into the cells. They can also induce cytolysis through a process through cytolytic granules called granzymes. And we have actually shown that the TREGs can actually express fast ligand and bind to fast on, say, for instance, microglia cells and lyse those microglia. That's one way that it gets rid of inflammatory cells. So it can be very important. Well, here's kind of the lowdown on 20 years of research on one slide. We showed that these regulatory T cells can cross the blood-brain barrier into areas of inflammation. And what happens is they'll encounter this inflammatory microglia, and it will react with these anti-inflammatory cytokines and drive the ROS and INOS down, and it will drive it over into either a resting or an M2 type of microglia. It will attenuate the inflammatory region. It'll suppress the inflammation. Well, it not only does that, but it also seems to react with astrocytes. And when it reacts with astrocytes, it causes the astrocytes to produce neurotrophic factors. Things like, you've probably heard of this, GDNF and BDNF, which actually protect the dopaminergic neuron. Now, that's kind of how we feel that the regulatory T cells work. We've noticed that there is another type of T cell, and this is called an effector T cells. These ordinarily are good T cells, but in the context of neurodegeneration, these are kind of the bad guys. And what we found out was that in our animal model, which we use MPTP, we can have the alpha-synuclein that I talked about that's misfolded and nitrated to actually leave the brain and go out into the cervical lymph nodes. And in the cervical lymph nodes, it activates the antigen processing mechanisms, the antigen processing cells, if you will, to present this nitrated alpha-synuclein to the T cells and induce those into effector cells. So now then you have this nitrated alpha-synuclein, again, which is nitrated, and it doesn't see it as self. Now it sees it as non-self. It sees this nitrated alpha-synuclein as foreign. So when it goes in and is presented alpha-synuclein, or this nitrated alpha-synuclein by these microglia, it reacts with them with pro-inflammatory cytokines, and it drives the microglia towards the red line into this activated or bad microglia. And so it has also, and of course, these microglia are the ones that are responsible for neurotoxicity. But it can also actually directly use these pro-inflammatory cytokines to interact with the neuron and directly kill that. So there's ways that we can induce this. I'll get to that a little bit later. But the important part is these activate or these suppress each other. The regulatory cell suppresses this, and the effector cell suppresses this. It's our hypothesis that these regulatory cells do a pretty good job early on in the disorder in Parkinson's. However, with age or with ongoing disease, these cells become exhausted, and it allows the effector T cells to take over and slowly to drive this reaction into the red line. Okay? Well, one of the questions that we asked, that's going to probably be most of the science part of it, but we then asked the question about what do the T cells look like in Parkinson's patients. And we did a clinical study. This wasn't a trial, but a study. And we were wanting to look what the T cell numbers and phenotypes were in Parkinson's. Are these regulatory T cells, in which we call TREGs, functional? And are these immune alterations associated with clinical outcomes? Well, we looked at 72 Parkinson's patients and 65 caregivers. Each one had their caregivers, so they were age and environmentally matched. And this is kind of the cohorts. There was 54 of the Parkinson's patients were male. 71 were Caucasian. They were mostly 72 years. I'm sorry, that's the number. Excuse me. Yeah, that's right. Okay, I'm right. So they were mostly 67 years of age. Their caregivers were 64. They had the disease for about 4 years. Their H&Y stage was 2, and their UPDRS-3 score was about 25.
So Dr. Mosley is the host?
Yes?
Oh, sorry, Dr. Mosley. Continue.
So what we found out was we found a set of effector T cells. These are the ones which we think are bad T cells. And what we found out was that when they are increased in number in the blood, that we find as they increase, they are associated with higher UPDRS scores or worsening UPDRS scores. The other thing that we found is looking at a TREG assay, and this is a little confusing. If we dilute out the TREG cells and ask if they can inhibit a certain proliferative response, we find out that the caregivers at each of these dilutions do so equally well. However, if you compare those to the Parkinson's disease patients, you find that their ability is far below this line. So they are actually losing the capability of inhibiting this proliferation or inhibiting the inflammatory response. And to kind of boost that, we found that one of these subsets of the regulatory T cells, of the TREGs, as they decrease or diminish in number, in percent, the UPDRS score increases. So it may be that these numbers, these type of TREGs are very important. So what we wanted to do, I gotta put this. Oops. Yes, I can. Okay. I'll just move that. There you go. All right. So we knew that there were some modulatory agents that we could use. We wanted to find one that we could use in the clinic, which could induce TREGs or increase TREG function We wanted one that would attenuate inflammation, and we wanted one that would affect neuronal activity or motor function. And there are a lot of these reagents out there, these drugs, which affect regulatory T cells. We have used glatiramer acetate. This is the one that I really won't talk about. Interleukin-2 is one that we're actually looking at right now, at least in animal models. We've also looked at vasoactive intestinal peptide, which we have produced with a company called Longevity Biotech, a VIP receptor agonist, which seems to work very well in animals, but they're having trouble finding a way to orally present this. And then the other one that we've been looking at is granulocyte macrophage colony stimulating factor, or GM-CSF. And it has actually been used also in Crohn's disease trials. Now, in our mouse model, when we use GM-CS, we do find that this, of course, does induce regulatory T cells, as you can see them increasing with increasing dose. In our MPT mouse model, where we have a increased amount of inflammatory response, that's what that is. When we treat with GM-CSF, it reduces that response. And if we look at the amount of neurons lost, this is in the MPTP treated animals, but if we treat those animals with GM-CSF, we can spare about, it's probably about 90 to 95% of those neurons. Okay, so we wanted to then look at a phase 1 clinical trial. And
Doctor, is it possible, can we have one moment? I just know, since we're sharing your screen, that seemed like a really good spot to take a moment and maybe check our waiting room, if you don't mind.
Okay.
Okay, we'll just take one moment to check the waiting room, and because that seemed like a really good spot.
I'll take a drink.
Okay, there we go. Oh, there we go. We had a couple more people. Wonderful. Thank you for joining us. We just, we're with Dr. Mosley on behalf of the Grace Strong Foundation. The doctor just went over sort of the first part of the clinical trials that he did, and he was just mentioning a treatment that was also used in Crohn's disease. And doctor, I'll give it back to you.
Okay. So we actually wanted to, the rationale for using this was we wanted to have a compound or a drug that could induce TREGs, or at least boost TREG function to affect clinical outcomes. And GM-CSF is one of those that has been used. It goes under the proprietary name of Leucine. Sanofi used to make this, but they sold it to Partners Therapeutics, who's handling the drug now. Its generic form is called sargramostim. But what it's been used for usually is in cancer chemotherapy, where people have they will treat them with either G-CSF or GM-CSF to mobilize bone marrow stem cells into the blood. They'll take those stem cells from the blood, and then they'll give them a very high dose of cancer chemotherapy, which not only kills the cancer, but it kills the bone marrow. So then they take the stem cells, which they have collected, and reconstitute the bone marrow with these stem cells. Now, sometimes it doesn't, you don't get that started very quickly. You don't get platelets revved up very quickly, or neutrophils very quickly. And so they will also give this GM-CSF in case to reconstitute those people. Well, this has also been used for acute radiation syndrome, for instance, like Chernobyl. Some of those patients were irradiated horribly, and they were able to rescue them using this GM-CSF. And of course, I mentioned that it's been used in IBD. So we wanted to look at this in Parkinson's patients. We wanted to test it for safety. We wanted to look at the immune profiles and TREG activity, and we wanted to look at its motor function. So this is what we did. We had 20 Parkinson's patients. 10 we randomized to placebo group, 10 we randomized to sargramostem. We looked at a baseline data, looked at baseline data for 56 days from week -8, 0. We treated them for 56 days, either with the placebo or with 6 micrograms per kilogram per day subcutaneously for 56 days. Then we stopped the medication, and then we evaluated these at 28 days after. So that's the design. This is the cohort. They had a mean age of somewhere 67 and 62. They had 7 years since their first symptoms. They had been 5 and 6 years after being diagnosed. They had UPDRS Part 3 scores of 24 and 20. They were 8 males and 2 females. All were Caucasians, and they had similar histories with respect to pesticides. So what was our adverse events? Well, it didn't matter if you were in the placebo group or sargramostem group, just about everybody had a adverse event of some sort. May have been very little. But did we have adverse events that leading to withdrawal? Yeah, we had 3 severe events and 1 serious event. And those were down here. One was a vasculitis, and it turned out this was our first patient. The pharmacy was actually reconstituting the lyophilized sargramostem with a water that contained benzyl alcohol. So this was actually a reaction to the benzyl alcohol. We had 1 patient that had an allergy response to the drug. 1 patient had chest pains. Just about everybody has chest pains with this, or bone pains. So it was too intense for her to take, so they withdrew. And then 1 patient had a small stroke. It affected their vision. They looked at his MRI and found out that he had been having these little mini strokes all along. So they didn't think that was due to drug. What were the other adverse events? Well, everybody that received sargramostem had abnormal laboratory values. They had increased WBCs and increased eosinophils. This is just a known function of this drug. It increases white blood cell counts. They all had injection site reactions. These are very mild reactions. As you can see, the placebo group that was getting saline, they had, the ones that reported them, had a severity of anywhere between mild and moderate, and so, as did the sargramostem group. So these are somewhat mild. I think you can put ice on them and they generally go away. People reported bone, well, they call it bone pain, but it's pain in the upper extremities, and that was a significant adverse event. They also report sometimes having chest pain because this is a bone marrow rich area up here in the sternum. The severity of pain was a little bit more, even more so than they received from the injection site reaction, at least how they guarded it, or at least they reported it. The severity of pain, of adverse events, actually was 1.6. It was somewhere between mild and moderate, or 1.2 for the placebo group. So that's the adverse events. This is looking at the white blood cells. You can see the peak, it's up to about 26, 28,000 per microliter. This is the neutrophil group. Those are increased. Once you stop the drug, they drop back down into normal levels. Again, start here, and the monocyte will go up, stop those, and those go back down. The real biggie was eosinophils. Eosinophils, this is very, very large increase for eosinophils. But as soon as you stop the drug, they return to baseline, to normal. Well, what had happened with the immune cells? Well, these effector cells that we talked about, it had no effect on the effector cells. They didn't increase, they didn't really decrease significantly. However, the sargramostem increased the number of these regulatory T cells, our good T cells, our TREGs. No matter if we looked at them as CD4 positive, CD25 positives, or if we looked at them with respect to FOXP3. And I probably forgot to mention that from the first slide. I'm sorry. FOXP3 is known as a transcription factor. It's a master transcription factor. This is a nuclear factor that controls just about everything that a TREG does. And this is the only transcription factor that is expressed, well, it's only expressed by regulatory T cell, by TREGs. So it represents a pretty good marker for marking regulatory T cells. The only problem is that it's internal, so it's intracellular. So we have to permeabilize the brain and get antibodies into the cell to actually get a grip on this, to see this. Well, another group of TREG population subsets were those which were expressing CTLA4. And these are particularly suppressive, as are these CD39 positives. Remember, these CD39 positives are the ones that are responsible for converting ATP into adenosine. So those are upregulated. Well, what about TREG function? These are pre-treatments, but here we have all of the PD patients together, and we did run a set of caregiver controls. So the question that I had here was did this loss of TREG function hold up in our study? And yeah, it did. It's not really significant, but you can see the Parkinson's patients' activity are below that of the control group. So yeah, their TREG activity is somewhat lower. Now, if we split the PD patients into placebo and sargramostem groups, we find that there's really no difference, no significant difference in these groups. But if we treat them with sargramostem, then we find their activity in these PD patients that climb up very good, significantly so, compared to the placebo, the saline treated group. And finally, even after post-treatment, 28 days after we stop the drug, we find that the sargramostem treated group still has TREG activity, which is significantly above the placebo group. So that's a good thing. And now we then looked at the UPDRS Part 3 scores. we actually included this as part of the safety measure because if we felt that if there was adverse events, there was very, very significant adverse events that they would show up in the UPDRS score, and we could stop the study if we had to. So what we found out, though, was it is pretty good marker of improved activity. Here is where, this is the baseline right here, and we start the drug at this point. And if you'll notice, both groups fall in the UPDRS scores. Of course, this is actually the change in UPDRS score. So if we're going below 0, if we're going negative, that's an improvement in score. And you'll notice that the even the placebo group is going down below. Well, we attribute this to placebo effect, and Parkinson's patients are known to it's a dopamine thing. It's a reward type phenomena. So they've been known to exhibit quite steadily this placebo effect. However, at week 4 after drug, we start to see that the placebo group goes back to where its normal levels were, and the sargramostam treated group still continues to go and fall below the baseline. And then once it's stopped, everybody goes back to the same place. So another activity that we wanted to measure, assessment, was looking at motor activity. And we did this through a magnetoencephalography instrument right here. And what they do, they put a cap of sensors on you, put them in this big magnet, and look at the magnetic field that is being given off on participating neurons in the brain. And that's what you see here. This group had been working with Parkinson's patients and had found out that if they do this finger tap exercise that they were looking at, that they had an event called a beta event related desynchronization. And I can't explain that because I'm not a MEG person, but you found deficits in Parkinson's patients in places like the premotor cortex, the supplementary motor area, and the precentral gyrus. And these are all areas that you would expect to be energized or giving off magnetic fields if they were doing movement disorder. And this, again, is looking at four pre-treated patients and looking at their, this is their beta event related desynchronization, or their ERG, and these are very diminished from what normal patients do. But what you do after sargramostam treated for 8 weeks is all of these areas are increased in size and activity. So that really says that the motor activity is increasing in these brain areas. So this was the phase 1A trial, the first one that we did. We felt that sargramostam was fairly well tolerated. There was mild adverse events. There were 4 serious or severe adverse events that led to withdrawal. The TREG numbers increased and the TREG activity increased after drug was ceased, that the TREG activity remained at least for a month. But the TREG levels returned. There was significant changes below baseline in the UPDRS score, some improvement, if you will, and motor activity increased beyond pre-treatment by MEGs. So our next step is probably the one that we're actually working on right now. It's a phase 1B trial where we're actually concerned about trying to get the adverse events lower and to increase the study time, see how well it can be tolerated. And that's what the phase 1B trial is. Is there any questions so far? Okay. So for this one, we're looking at starting on day 0 with leucine. This is going to be, instead of 6 micrograms per kilogram, we're going to use half that. We're going to use 3 micrograms per dose. We're going to have 5 days on and 2 days off. And it's an open label. Means everybody knows that everybody's on it. But the problem is that we have very few. We enrolled 5 males and 1 female. She was excluded because they couldn't get enough blood from her. She had venipuncture problems. They had similar environmental exposure histories with respect to pesticides and chemical fumes. The average age was 64 years of age, and the average years of diagnosis was 8, somewhere between 3 and 15. So what we're going to do is we're going to take monthly assessments from 2 months before the injection of the drug, and then 6 months after, and then every 2 months after 6 months, so at 8, 10, and 12. Now this study can be extended to 36. What I'm going to present today are the results for just a year of this. That's really all that we have worked up. We're also going to, in baseline and at 2 and 6 months, we're going to do what's known as a leukapheresis. Leukapheresis is where you take out the white blood cells in one arm, well, you take out all the blood, and then you basically eluterate the leukocytes, the white blood cells, and put the red blood cells back in the arm. So you can get a lot of cells. And we needed to do this in order that we could look at methylation analysis and transcriptomics and proteomics analysis for the white blood cells, for the regulatory T cells. I'm not going to talk about transcriptomics or proteomics. I'll talk a little bit about the methylation process. Okay, so adverse events. Well, again, this is a representation of the adverse events, severity and category of adverse events for the phase 1A. This is the phase 1B that we're talking about right now. So we have a less number of phase 1Bs, and that's actually, you can see there's also fewer adverse events per subject. There's fewer than it was from the phase 1A. There's fewer numbers in the phase 1B. And if you actually take into consideration that this is 12 months and this is 2 months, you can see that in the phase 1A study, you had about 7 or 8 adverse events per month. But down here in the phase 1B, we may have 1 or 2 per month. And overall, the severity of these, whereas remember it was 1.6 severity for the phase 1A, this has dropped down to about 1.5. So the numbers of events are less and the severity is less. What are they? Well, again, looking at the category, again, we still have abnormal lab values a little bit. We have injection site reactions still, but then the the chest pains and the bone pain in the extremities have vastly gone down. What about what blood cell count? Well, we had changes in the white blood cell count, in the percent of lymphocytes, the percent number of monocytes, and the percent number of eosinophils and the number of basophils. And this is just to show you the two of these total numbers. We have an increase in the white blood cells. There's really not a big increase along here. It's just at these 2 points for the white blood cells. For the neutrophils, there's again at 2 months and 6 months, and then at 6 months for the monocytes. And for the eosinophils, again, this 2 month and 6 month kind of events. These are the only ones that we had significant increases. Everything's a little bit longer. In the metabolic panel, we had changes, significant changes in the AST and the protein and albumin in the blood, and in the anion gap and the CO2. But the blue is the typical ranges where they typically fall. And while they're significant, nobody's falling out of the ranges that are considered normal, except for the AST. And for some reason, the AST actually dropped below. You would normally expect if you were having liver damage that this would climb way up. Why it goes way down, I haven't heard an explanation for that or found one. Okay, UPDRS. Well, this is UPDR part 3, and this is for each of the individual subjects. This is their UPDRS score and course. So it's a little bit hard to follow. So if you group these together, you can see that by starting at 0 is where the drug starts. Probably it keeps falling down to probably about month 4, and then it kind of levels off. None of these are significant statistically because you have such a variability here. And that's why we actually like to look at not at the total UPDRS score, but how it changes from the baseline. And that's what we're looking at. This is each individual's baseline changes or changes from the baseline. This is the actual baseline for grouped together, starting again at 0. By week 3, we've dropped to at least about -5, -4.5 improvement in the score. It's not significant yet. Again, we have only 5 patients, but it really takes until it drops to probably about 7 points improvement that it really begins to be significant. What about, this is not UPDRS, I'm sorry, this is T cells. We see that the T cells are basically increased in CD3 positive T cells, CD4 and CD8. This is percentage. With the number of lymphocytes, we see an increase in the total number of CD3s, total number of CD4s and CD8s. What about the TREGs, the good T cells? Well, yeah, they kind of go up significantly so by, I guess, by 3 months, and they stay up for a while, and then they start to go down, and they're kind of at this point for a relatively long time now. And what about the bad T cells? Well, for some reason, they went up when we first started these 5 patients. We didn't see this in the first study. We're not sure why this is. We have no explanation, but they tend to fall and they kind of staying around this level. There doesn't seem to be any significant increase. TREG increases, TREG and TREG subset. Well, this is the FOXP3, and you can see that it's significantly increasing up as we go along. So that's a good thing. The other thing is this are Helios positive TREGs. We're seeing an increase in the Helios positive TREGs. This is another more stable, more reactive, more active type of TREG. CTL4s they have a little peak that may be associated at this 2 and 6 time. But the interesting one is the B7 integrin positive tregs. These go up and pretty much stay up. And we're very interested in these because the integrin beta 7 is one of the surface markers for which these cells find the endothelial cells for which they can cross into the blood-brain barrier. So this is one of the integrins sees the adhesion cell molecules on the endothelium, and it can then cross the blood-brain barrier into the brain parenchyma. Well, what about TREG activity? Okay, so this is the baseline activity, this blue line. And after 2 months, it peaks up here. It drops after 4 months to here. It drops after 6 months to this dark green line. It drops to 8 months to this brown line, 10 months at this black line, and 12 months it's actually increasing up to this aqua line. Now, everything is above this, and it's significantly so. You can see the p-values for the elevation. So this is significant increase. I told you about the FOXP3 and demethylation or methylation. This is why we were doing the leukapheresis, is that we needed to isolate lots of tregs so we could get their DNA and look at the methylation. Why is methylation important? Well, the more methylated the drug or the DNA is, the harder it is to read the DNA in order to transcribe the messenger RNA for the protein. It's harder to express this protein. So the more demethylation it is, the better that you're going to have this FOXP3, this FOXP3 protein expressed. And that's what's shown here. As we find in baseline, we find it somewhat lower than what it is when we treat with sargramostam. This is about 65% demethylated, but if we treat with sargramostam, we can boost that demethylation up into about 80%. And we find out that the activity is indeed associated with good TREG activity. What I'd like to do is to kind of, I guess, explain, we wanted to look at correlations of some of these and about how it relates to TREG function and TREG activity and UPDRS, but it's hard to do in this kind of format, just looking at a line and doing correlation. I'm sorry if you don't get that. What we had to do is we had to take this number, this line, and convert it into one number for each line. And that's what I'm showing you here, is one of the ways that we can do that is by looking at the area under the curve. And that's what we have done here. And the other way that we can do that is looking at the line and asking, okay, well, how many tregs does it take to get up to 50%, to a standard 50% activity? And if you'll notice, the one with lower activity, it takes 67.2 times 10 to the third to get up to that level. But on one with higher activity, it takes 21.7 times 10 to the third. So it's a way of looking at the activity that you have, two ways of looking at the activity of TREG. And that's what this correlation is actually telling us, was that there is a very good correlation between both of these measures of TREG activity. What we wanted to look at was the correlation of TREG activity. And this, we're going to look at the area under the curve versus the TREG numbers, the TREG percentages. And if we look at the TREG percentage or any of these, we find that they are directly correlated. I think all of these are significant. Yes. So we see that the TREG activity, the number of TREGs, the percentage of TREGs, if they're there, they have good TREG activity as their percentage increases. So does the FOXP3. These B7 positive TREGs, as they increase, their activity increases, and this subset of TREGs and this subset of TREGs and this subset of TREGs actually represent very stable and mature type TREGs. So as their subsets increase in the blood, so does the TREG activity. This is just another way of looking at it, and I won't bore you about it. I just want to look at these are basically showing the same things, the same subsets, except for one. What this is looking at is looking again at the number of TREGs to get to 50%. So again, the lower number of TREGs represents a higher activity. So with the higher activity, you find that they have a higher the higher number are, they have a higher activity here. The lower number of these define the lower number. Okay, so that's all I really wanted to say on that. Oh, I'm sorry. The other thing that I wanted to say is these are the CD, that I was able to pick up this CD39 positive subset of TREGs in this population using this method. So what we really wanted to do was ask, how do these correlate? How do these TREGs and TREG activity correlate with UPDRS Part 3 scores? And again, looking at this 50% inhibitory number, the lower number represents a higher activity. And indeed, with higher activity of TREG, the lower your UPDRS score is. The higher it is, the worse, the less TREG activity is, and the higher the UPDRS score is. Similarly, if we look at this area under the curve method, remember that the area under the curve is the lower the area under the curve is, I'm sorry, the higher the area under the curve is, the higher the activity is, and the lower your UPDRS score is. The lower the activity, the area under the curve is, the lower the activity, and the higher the activity, higher the UPDRS score is. And so these are basically showing that this B7 positive population is looking at if the higher percentage of, if you have a high number of these B7s, you tend to be correlated with a lower number of UPDRS score. But the lower that these exist, the lower percentage that they exist, the higher. And that goes along with all of these different ones. Typically, the higher percentage that they are, the lower they're correlated with a lower UPDRS score, whereas the lower the subset is found in the blood, basically the higher the UPDRS score. So what we get from this so far, if it holds up, is it's safe and tolerable. We think we can do extended dosing. The frequency of adverse events and the severity is diminished using this dosing scheme. The improved, we showed that sargamstam actually improved the UPDRS Part 3 score from the baseline. It increased TREG and TREG subset levels. This correlated directly with TREG activity and indirectly with UPDRS scores. And the TREG activity was elevated over baseline. That correlated with lower UPDRS scores. And we had an increased demethylation of this TREG specific demethylation region that allows FOXP3 to be transcribed and for regulatory cells to be more active. So the next step for this will probably be, we're in talks right now about doing a phase 2 trial with 240 patients and 2 doses and a placebo. So that's kind of all I had. I'd just like to thank everybody, acknowledge everybody that was in my lab and Howie Gendelman's lab, especially Kate Olson. She's the one that's is responsible for doing a lot of this current one. The MEG lab with Tony Wilson, our flow analysis facility, and of course, our CRC nurses and personnel, which get the patients and keep the patients scheduled. Our neurologist, Pam Santa Maria, and Danish Bata, and of course, most of all, our patient and PD patients and caregivers. We have a variety of funding support for this, community support. The Francis and Louis Bumpkin is a big benefactor. These are the people that own the Nebraska Furniture Mart. I don't know if you've heard about it. Of course, Sanofi and Partners for including the giving us the drug, and of course NIH grants for which we work off of. So that's all I have.
Okay, that was great. Thank you, Dr. Mosley. Does anyone have any? Oh, there, Eric, I'll pass it back to you.
I was on mute till you started talking. I was like, I'm not on mute. Yeah, it was very informative. I have lots of questions, all that I need to refocus in my head. Yeah, that was definitely very informative. I know some people felt the same way, messaged me throughout the lecture, that they were trying to come up with questions. Wow. So basically the goal, if I translate it right, the goal was basically to activate the T cells to help transmit the FOXP3 in order to get low UPDRS scores.
Well, that's kind of the way we look at it. It's really to activate it. One of the things that we think is happening is that once we are able to induce TREGs and or boost their activity, we don't really know what's really happening to these TREGs. We don't know if they're just dysfunctional. That's kind of my bias, is that because we don't see a lot of loss in the TREG numbers. There may be a difference in TREG numbers in the subsets, but they tend to be less dysfunctional. So what we're trying to do is either boost these ones that are there, boost them function up, or generate new ones out of the bone marrow and have those then expand and do the job. So it's basically utilizing GM-CSF. We think one of the ways it works is by allowing the demethylation of these the gene right or the area right before the FOXP3, allowing more FOXP3 to be produced. And FOXP3 is the transcriptional factor that goes and kind of controls all of the other things like the anti-inflammatory cytokines and the CD3 expression and CTLA4 expression and CT39 expression. So we think that this FOXP3 is made more stable and more expressive if with the GM-CSF.
Okay. And then I was jotting down notes and trying to keep up. Did you explain the importance of the high UPDS scores versus the low?
Well, the higher the UPDRS score is, the more severe the movement disorder is.
Okay.
So that's the deal with the UPDRS Part 3. So the higher your UPDRS score is, the more severely your motor disorder is.
And that's where the importance of those last slides that you were showing, as far as the higher TREG scores versus the lower UPDRS scores, and vice versa.
Right. Usually we find that if the TREG activity is high or the TREG number is high, that is associated with a lower UPDRS score. The lower the TREG numbers or the lower the TREG activity is, we find it associated with a higher or more severe type of motor dysfunction. Now, we don't know that's a correlation. We don't know if there's a cause and effect. That's just a correlation.
Chris, you had a question?
Yes, and Dr. Mosley, thank you very much for presenting this. It's been a while since I've had some microbiology classes, so I'm still kind of processing through some stuff. And if the question, it popped in my head, so if it doesn't make sense, just let me know. So what other inflammatory issues could correlate with this? I mean, so I think where I'm going is, and I spoke with Eric about this earlier, so we know the connection between the digestive system and Parkinson's.
Right.
So is there any other inflammatory conditions not specific to the digestive system that could be related, you think? Or
well, the ones that we're talking about are basically inflammatory conditions in the brain.
oh, okay, gotcha.
That's where this is all.
It's all okay.
So the microglia, wherever you find deposits of alpha-synuclein, misfolded alpha-synuclein, you find damaged neurons in the substantia nigra and the striatum. They're always kind of find these microglia or inflammatory cells. I mean, that's what they do. They gobble up debris and damaged cells and get rid of them. The problem with Parkinson's disease and a lot of the other neurodegenerative diseases, they don't turn off. They don't shut down. Usually they do their job, they get rid of the damaged cells and they go back into the resting state, and you don't see from them or hear from them until some other damage occurs. But in Parkinson's disease, they don't turn off. Now, that might be because there's just so much damage going on. There's so many neurons that are now being affected that they never can get turned off. But as long as they're going, they're probably going to cause more neurotoxicity and more neurodegeneration. So the point of this, hopefully, is what we can do. I mean, these neurons, once they're gone, they're gone. And so in Parkinson's, once you become diagnosed, you're probably lost 50 to 70% of the neurons. So what we are trying to do is come up with a way to attenuate the inflammation and at least interdict the neurodegeneration as much as possible. And hopefully somewhere, somebody will come up with a way to do neuron replacement, for instance, like was done back in, I think it was the 1980s, I think. So those studies, they kind of look promising. I don't know why they're not being done more now, but
it's kind of putting all of the little pieces together to get.
Yeah, I guess the other thing is this thing about alpha-synuclein and Lewy bodies and things like that. And so you've got this protein that's getting misfolded all the time, and it's getting nitrated because of this inflammation, and it's kind of feeding itself. So once you get that, if you get that under control, you may be able to clear some of that out and stop the inflammatory response. That's kind of another way to do that. There's several studies going on, clinical trials from a group in Austria, I can't remember, AGFS, who they're making mimetopes to alpha-synuclein or nitrated alpha-synuclein to give as an antigen. And hopefully the person will make antibodies to the alpha-synuclein and clear the alpha-synuclein out of the brain such that you can no longer get this misfolded protein, which is activating all the microglia. So that's another way that people are actually doing it.
Very, very interesting.
Yes, thank you.
Thank you very much. Anyone else have any questions? And if you guys can't think of any questions now or mind are racing like mine has been for the past hour, feel free to write them in the chat. I'll post my email here and my social media so you guys can always submit questions. We'll have a follow-up interview on the podcast for Dr. Mosley. We'll post the date on that once we make arrangements.
Yes, so for more information, and Eric is putting the information in the chat, but it is the Gray Strong Foundation, graystrong.org. We do have some upcoming events that will help us support the Parkinson's community here locally in the Lehigh Valley and the tri-state area, and across the US. As you can see, we have doctors that help support us all the way in Nebraska. So so from here to Nebraska and in between, we do have a tournament, a cornhole tournament coming up in October, Saturday, October 16th. That will be at the Hanover Canal Park. We are in the midst of confirming a symposium with the Lehigh Valley Health Networks and possibly St. Luke's and Sacred Heart for April, as well as our annual Tricky Tray. You can find Eric at Trembling EMT, and you can find Dr. Mosley. Dr. Mosley, are you a social media fan?
I do not do Facebook.
So you can find Dr. Mosley and his, I could say his coworkers and the people that he works with for the research information at the University of Nebraska. Medical, wait, I know I have it. UNMC.
Right.
There we go.
Once again, thank you, Dr. Mosley.
Well, thank you for letting me talk. I appreciate it.
Definitely very informative. And I'm surprised I kept up with most of it. I was shocked. I shocked myself.
Thank you to everyone who joined us this evening. Please, if you know anyone or have family members or know of anyone with Parkinson's or their families or supporters of the Parkinson's, please let them know about our monthly lecture series and upcoming events, and let them know that they can come to us for their support.
I'll stay on for a couple more minutes if anyone wants to ask me questions or stuff like that. And Dr. Mosley, thank you again for your time and appreciate it. And we'll talk.
Thank you, Dr. Mosley.
Thanks, guys. Have a good
Thank you.
evening.
You too.
Hello all. How you guys doing?
Good.
That was like a buffet, like a Caesar Palace buffet.
I was in and out. I'll be honest. I was in and out. I was like, he got a little technical for me at points.
I know, but
A little technical. I mean, some of it, it's going to take me a while for some of it to sink in.
Right.
Yeah, I was having trouble trying to focus and listen to most of it too. So,
Well, that's the joys of statistics. Once that comes
Right.
into play, I mean,